immediately let's jump into uploading the data first, and because this will take some time. So I'm just going to first do the task and then I'll explain what I'm doing. And before we do that, I just want to make sure that everyone is up to date with what I'm doing. So I would suggest you write down the things that I'm telling you. So not the entire thing. I just want you to make notes of exactly what link I'm following or which tool I'm using, what are the parameters that I'm using, and so on. So this will be helpful when you try to do the analysis on your own. So straight away, yeah, I hope that everyone's logged in into Galaxy, by the way. So in the hope that you've already logged in, what I will do is I will go to upload data right here. So I'll be clicking on this upload data, and you will see this page, right? So once you see this page, you just go click on choose local files. And once you choose local files, you just have to browse exactly where you have placed all your documents, the attachment in the email, the one that says practical. So once you unzip that, you'll find all these data inside. Now, what I want you to do is just drag everything, select the entire thing. So once you've selected that, you just open it just like that. All right. Now, once that has been done, there are different options here. Like you can specify the name, you can specify the type of file this is. So this is a FASTQ file, genome. We don't actually need to do this right now. It's actually good practice if you do it, but it'll just detect automatically what kind of file it is. So we know it's a FASTQ file, and then the tool will automatically detect that it's a FASTQ file, and it'll change the labels accordingly. Now, genome, we're not going to specify that right now because we'll be doing alignment later on. So we'll just leave that unspecified. And once you have opened all that, what you will do is you're going to click on start, right? You're just going to click on start, which I'm not going to be doing because I've already loaded all the files, but you click on start and that's it. You just wait for the file to load, which will be displayed here in the status. So you can see the percentage or the speed at which the upload is taking place. And you just wait for that to finish. Right, hopefully everyone has done that. So while it's uploading, let me just give you a brief explanation on what the data set that we have taken is. So the demo data set that we're using right now, it includes 3 replicates of data for a breast cancer cell line. The cell line is specifically HC1395. That's the name of the cell line, all right? So it's a breast cancer cell line. And we also have 3 more replicates of a normal cell line. So the first one that we have seen is a cancerous one. And the second one that we have seen is from a normal cell that matches the one from the cancer. Okay, so basically what this is it's going to be a tumor versus normal cell line comparison. Now, the reads are paired end. So paired end means, if you remember, we're going to be reading one end of the read from the 5 dash to the 3 dash, and on the opposite side, again, we'll be reading from the 5 to the 3. So because of that reason, you see 2 files here. One is labeled as rep1 r1, and then the second one here is labeled as rep1 r2. So that means these are both the same replicates, replicate number 1 from the normal cell line. This is the read 1, and this is read 2. That's what it means. So the one that says rep1, it's actually not 2 files. It's just 1 file which has been divided into the 2 reads, and we'll be merging that later on to make it into 1 read each. And also the same goes for the others. So we have 6 files for the normal, and then we have 6 files for the cancer type. Now, just a general info on the type of reads that we have. So the paired end reads are 151 MERS, so K MERS. Maybe the bio people are not familiar with the term, so it just means that the DNA from where we have sequenced, we have cut them into small strands of 151 nucleotides each. So that's what it means, 15 MERS. And then this was generated by using Illumina technology, and the instrument is HiSeq. That's what has been used. So Illumina, as you remember, it's a second generation sequencing platform, and it's used for generating short reads. So that's what they have done here. And the test data that we have, so these have been pre-filtered to a certain extent, but we'll be doing some slight more tweaking later on. And obviously, you must have seen that the length of the sequence is quite small. So the reason behind that is, if you take the whole entire genome, 3.2 billion base pairs, it's going to be a really large file. It's going to be approximately some, 2 to 3 GB, depending on the depth. So the more, like, because of the content, the size of the file may increase. So this one is taken only from chromosome 22, mind you. So in the end, when we run the data and we see the results, uh, don't expect like a really big output with some really significant genes that you see in cancer and all that, because right now it maps only to chromosome 22, which means it'll show you only those genes that are present in chromosome 22 and nothing else. So yeah, let's wait for another couple of minutes. The next thing that we're going to do, so I just have this complete Galaxy workflow, just so that everyone is in the same page. Right now, what we have done is we have taken the samples, the normal, the first replicate, the second replicate, cancer, first, second, third. Yeah. Now we have 2 FASTQ files. One is for the first read, for the second read, first read, second read. Now, what we're going to be doing is we're going to be running FastQC. That's the tool for checking the quality. And we're going to be running it on all the samples. Okay. So to do that, in the search bar, simply we're going to type FastQC. Okay. So you might want to write that down in case you forget the steps. So once you upload the file, which I hope you remember, we're just going to look for FastQC in the tools, and we're going to open FastQC. Okay. Now, there's a list of options here. We can, what we can do is first, here it says single data set, right? So the input for this is just one single data set. It's going to take only one input and it's going to give only one output. So instead of single, since we have 6 different files with us, we're going to go for the other option, multiple data sets. So we're going to click on multiple data sets, and you get something like this, right? So once you click on that and you get this option, yeah, I'm just going to click on the first one, and then I'm just going to press Shift, and I'm going to select all of those. So directly click on one, press Shift, and select all the replicates. Yeah. So all the normal ones have been selected. The same thing, you just keep on doing that. Press Control 7, and then till 12. So total. Oh, sorry. Let's try that again. We'll select the first one, and let's just directly go to 12 by scrolling up, Shift, and select. There we go. So I hope you followed that. We've selected all 12 samples now. So 6 for the normal, 6 for the tumor type, right? Of course, you can always just say single data set and you can just keep on running, but just make things faster. That's what we're going to do. Now, for this workshop, just to keep things simple, we're not going to specify anything here. And you can directly go, scroll down, and we can run the tool. So just click on Run Tool. That's it. So let me quickly recap the steps in case anyone has missed that. We're going to look for FastQC, open FastQC, and we're going to select multiple data sets. And after that, we're going to select whatever data set we have, the fastq.gz. So it's just a zip file, just to make things smaller, compress the file. And then once we're done with that, we're just going to select it and we're going to run the tool. That's it. So I believe this will be relatively faster than uploading the data, which certainly takes a lot of time. But yeah, so this is the kind of output you will get. Here it just says FastQ on data, so and so, right? So there are two types of output files that will be generated. So on data 30, 30, that means this is for the first read of this one. I mean, this is the name of the file. And this is the output for the first one. It'll automatically tell you which data it is. Based on this, you'll be able to find out exactly which data or which data set was executed. So the first one, it's in HTML format. It's actually a visual output. So you can visualize the output through that. And then you have a raw data format, which will give you everything in the form of a text. So it'll generate that for all the files. So let's take a look at one of these. So this is the kind of output that I showed you before. Yeah, maybe I'll just, yeah, I think that's better. So we see that the quality is quite okay-ish. It's not, below 28 or 30, as a matter of fact. So anything below 20 would have, done something to that. Some of these outliers are falling in this region, but we'll still consider that it's not an issue, right? And then these are things that you shouldn't be concerned about that much right now, but we'll take a look at that later on. Now, per sequence quality score. Remember I told you that the per sequence quality score should be something like this. It should be a bell-shaped curve skewed towards the right side. So that means all the files should have a Fred quality score of something greater than 30. So this one seems to be pretty okay. And of course, you can always check if it has passed the test or not. So simply by clicking on this, you'll find out if things are okay or not. So remember I told you that the length of the sequences were 151 mers. So there you have it. It tells us that the length of the sequence is 151. And here you can see here positions in the read. So 150 to 151. That's the overall length of our sequence. Now we've gone through this. It says okay, and there are 3 of these. Exclamation means it's a warning. X means it has failed. Okay. Now let's take a look at the 3 output where FASTQ report says that we have failed those tests. So the first one, obviously, as discussed during the workshop, it's the per base sequence content. Now I already explained this to you. In the case of RNA-Seq, you don't have to worry about that because I said around 10 to 15, the first 10 to 15 nucleotide sequences will be quite erratic. So it's very close to 15. It's between 10 and 15. So we're just going to leave this, nothing to worry about. And we'll move on to the next one, which is the per sequence GC content. Again, as discussed earlier, this will deviate slightly from the actual theoretical GC distribution. Okay, so initially during the workshop, I showed you only these two output mostly because these are the things that are concerning when you look at the output of a FASTQ report. So I'm telling you, these are nothing to be concerned about. This will generally happen in RNA-seq experiments. So you can leave that aside. The rest seems to be okay. Now, we can more or less ignore the warnings also because sometimes because of all these issues, you might get warnings, but that can be ignored for now. But let's take a look at the adapter content because this is something that shouldn't happen. Now, look at this. We see that the pink line, it's quite straight, right? So what it'll show you is Galaxy, or rather FastQC, has a collection of the type of adapters that are generally used during sequencing. So if it finds that there are some certain adapters that are still attached to the sequences, so it'll try and warn you about all those. So all the available ones are mentioned here. And if there are no adapters, you just see a smooth line, straight line like this. So example, solid, you can see that it's quite straight. And I'm sure that underlying all underneath that layer, you have all this Illumina small, Illumina transposase sequence, Nextra, sorry. So the problem is Illumina universal adapters. We can see that we have adapter content that goes up right here, right? So how do we deal with that? Now, the one topic I didn't cover during the workshop is because I wanted to show you hands-on what can be done when you have adapters or when you have some sort of contamination happening, right? Now, we can simply remove the adapter. Speaking of which, I hope everyone has generated their FASTQ output. Okay, so what we're going to do is we're going to look for a file to do some trimming for us. Trimming could be anything. It just means in case you have, if the yellow bars are extending all the way down, we'll use a sort of scissor and we'll just smoothly trim it so that it'll fall below a threshold that we specify. Okay, so if we want to correct the adapter, let's take a look. We see that it falls within around 120-ish, around from 120 till the rest of the sequence. So what we're going to do is we're going to remove around 20 base pairs of the end of this sequence. We don't want to over trim it because that could mean loss of data, and that is something which we don't want. So we're just going to remove around 20 base pairs towards the end. That means towards the 3' end, using a tool called Trim Gallery. So you can write that down. Trim Gallery is the tool we'll be using. Of course, there are several other tools besides Trim Gallery. There's Trimomatic, there's CutAdapt, and depending on the situation, you can just change the tools and try to figure out which one has the best output or which one does the best job. For the time being, we're going to be using Trim Gallery. So we have looked up Trim Gallery in the search bar. We'll be clicking on that. And over here, we are going to select paired end. All right. So we know that the library we have, it's a paired end sequence. We'll be selecting paired end. And we are going to select multiple data sets again. Now this time, instead of selecting everything all at once, what we're going to be doing is we're going to be selecting only the R1s in each case, R1. So by clicking Control, you can just skip the R2s and select the ones that says R1 only. I hope you followed that. Now for the second one, obviously we're going to take the second read. We're going to select R2s only. Oops. There we have it. So we have selected the R1s and the R2s respectively. So R1 in the first one, R2 in the second one. Okay. Now we scroll down, we see adapter sequence to be trimmed. What kind of adapter sequence do we want to trim? We can go for automatic detection if we want, but we're going to go for Illumina Universal because that's what the output of FASTQ report says, that Illumina Universal Adapter is the one that is creating some problems in our data. So there we have it. And after that, what we're going to do is we have seen that 20 base pairs towards the 3-n, right? The percentage of adapter suddenly jumps up. So we're going to remove 20 base pairs from both read 1 as well as read 2. So you will specify 20 here and 20 here. It's going to remove 20 base pairs each from the 3-n of read 1 and read 2 as well. So this step, by the way, it can vary depending on the sample you have. You know, some samples they don't even require trimming at all. Some they need a lot of rework. So, but that usually, doesn't happen because that means there's something wrong, probably something wrong with the way you have done the sequencing. So right now in this case, we're going to go for 20 because that's what we want. So once you have done that, you will simply go and run the tool. So just a quick recap again. While the tool is running, we're going to look up trim galler, and in the library selection, we're going to select paired and reads, paired end option, right? Because we know that our reads are paired end. And of course, if you have single end, then you will just select single end. That's quite obvious. And reads in FASTQ format, the first one, we're going to specify only the R1s. And for the second one, we're going to specify only the R2s. After that, the adapter sequence that has to be trimmed, it's going to be Illumina Universal. And again, this you'll have to verify with the output that is being shown. If it's not Illumina, then you'll have to go for something else. Otherwise, if it's not listed here, then you can go for the automatic detection and make the tool figure it out by itself. If it's not, you can go for a different tool and see what it does. So here, what we're going to do, again, remove n number of base pairs from the 3-n of read 1 and read 2. So n number of base pairs is 20 in our case. So we're going to set it to 20 each. Then we're going to run the tool. Okay, I hope the trimming part is done now. What we're going to do now, okay, so you'll have a list of files generated there. It says stream galore on data so and so, right? Now, again, we're going to repeat the same process. We're going to do FASTQ check again. So simply search for FASTQC. And again, we're going to select multiple data set. And this time, what we will be doing is, instead of using the original sequence, we're going to be running this tool on the output that trim galler has generated. So select all the output that has been generated using trim galler, using the shift, and by selecting all the data, and we're going to run that tool once again. Okay, hopefully the FASTQ files have been generated now. What we're going to do is, by the way, this step is entirely optional, but just to make your output more presentable, what we're going to do is we are going to be combining all the report using another tool here. It's called multi-QC. Multi-QC. Multi-QC, yeah, that's the tool. So what it'll do is it'll aggregate all the results from the fast QC output, and it'll give you a pretty output with all the details included in that. So to do that, you're going to click on multi-QC, and here, which tool was used to generate logs? Obviously, it's not BAM tools, so you're going to click on the drop-down menu here. You're going to look for fast QC right here. All right. So you're going to click on that fast QC, and you're going to insert fast QC output. Okay. Now, once you do that, remember it's not the first fast QC output, it's the one that is generated after Stream Gather. So you're just going to take all the raw data, select only the raw data of the fast QC output. Okay. So once you have selected all that, of course, there are some options here. You can change the title of the report. You can name it whatever you want. You can plot different things. You can give different things as output. We don't want all that. We just want a plain simple output. So what we're going to do is we're going to run the tool after selecting only the raw data from the file that was generated after trimming. Okay. So you just click on run tool after that. So another recap, we're going to use multi-QC. We're going to select fast QC in the drop-down menu. So you'll have to search for that. You're going to select fast QC, and then you're going to specify the output, the type of output content that you want. So it's raw data. We have selected raw data, and then we simply run the tool. Okay, I hope that the output has been generated. Now, similar to FastQC, you will see two different files being generated. So as usual, we're interested in one that is in HTML format. It'll give you a nice graphical representation of the qualities. So yeah, by the way, I forgot to mention that if you want to see the content of a file, you just click on that file, it'll expand it. So I clicked on multi-QC there, and there's an eye icon right here that says display. So you just click on that. Yeah, there we have it. So multi-QC, it's basically the same thing as your fast QC output. It's just that it has merged everything together so that you can, when you have just a couple of data, if you have 2, 3 data sets, say, for example, then it's easy for you to keep on comparing them one by opening each fast QC output, right? But when you have a huge number of files, because you can have up to 12 samples or even more than 12 samples. So imagine you have paired end sequences of 12 samples. So you'll have 24 output. and so on. So depending on the size of the data, multi-QC makes it easy for us to view all the results in one page. So yeah, now let's directly look at adapter content. So yeah, this is like a sort of uh, status check. It shows you exactly like a summary of the entire quality. So per base sequence quality, we know that it's going to be in the red zone. Per sequence GC content also, we know it's going to be in the red zone. For some, it's in the green zone. That's well and good. Some are in the yellow, most are in the red, right? As for adapter content, you can see that it's all in the green zone. That means we don't have to worry about anything. All those adapters that we have seen are gone now, practically gone. But there's one thing here that says per tile sequence quality, and this is also pretty much in the red zone. And why is that? So just as an example, let me open one of these FastQC result in a new tab. Yeah. So this is what per tile sequence quality is, right? And you can see that there's a cross mark here, which means that it has failed quality check. Now, there's a reason for that. We have been ignoring these, but you know, during sequencing, each reads are loaded into something called flow cell. So now flow cells are a sort of platform where you will attach those adapters and the sequence will keep on adding to those, like it'll keep on extending. Now, sometimes what happens is air bubbles can get trapped inside flow cells, all right? And that's a very real, and it's actually a physical problem that occurs during sequencing. So it's not a contamination, but it's a technical flaw during the process of sequencing. So when these bubbles get trapped, it creates problems. Now, FastQC, what happens is it's quite rigorous. And then when evaluating the sequence, it may flag those samples just because the red specifically. So these are the tiles that create problems, and usually we would just ignore it. But since FastQ is quite rigorous, it may flag those as problematic. That's why uh, you you get this kind of output, and then it fails the quality check. But looking at the overall results of the quality check, we have already seen that most are good. The basic statistics is quite great, in fact. So we can ignore the part where the tile sequence quality fails. So we can move ahead and yeah, let's tally that with the flowchart I have. So we have the cells. For each one, we have performed FastQC. We have trimmed it using fast trim galler. We have generated FastQC again. We have aggregated the results into multi-QC. So the steps that we have done till now, that covers quality control. So we're happy with the quality of the data. Now we can move on to mapping and alignment. Now to do that, what we do is we're going to be using a tool called HiSat. Okay, so HISAT, that's the name of the tool. And we're just going to open that tool. And let's just, we're going to be using a built-in genome. So this is the source for the reference genome. Now remember, we need a reference genome and we need the sequence that we want to get it mapped to. So we're going to be using reference genome. And in the second option, it says select a reference genome. Now we know that we're dealing with human genome. So of course, there are several organisms here. Like I said, many organisms have been sequenced till date, and all those will be mentioned here. Like there are a number of reference genomes available, but we're interested in humans. So what we're going to be using is you can type HG38 in the search bar. And once you do that, you will see human December 2013 HG38. We're going to select that. All right. Now, the reason why we're using HG38 is because this is the latest version of the primary human genome assembly. Now, this is the official version that is being used by the UCSC genome browser, and UCSC genome browser is, I mean, it's the it's a standard. Uh, I mean, it has the standard for which we can use as a reference when we're trying to find any sort of genes, any sort of introns, exons. All the information is given in this browser. So this is what we'll be selecting, and it's always a good option to use the latest ones, because what happens is the previous version, which happened to be HG19. Okay, now HG19 and HG38, there's a difference because, of course, both are human genomes. It's basically the same thing, just that uh, it's actually quite different, because if you try to load an HG38 aligned output and try to view it where the reference genome is HG19, you will see that the genes that are actually supposed to be there on HG38, they're on the wrong location in HG19. So this is something you want to completely avoid. And whenever you want to work with any sort of alignment, it's always good practice to use the latest build of genome available, irrespective of whatever paper you're using, paper you're reading. They may report that they're using an older version of the genome, but like I said, good practice to use the latest version. So again, we're going to select parent sequence, right? We're going to select multiple data set for both file 1 and 2, parent, file 1 and 2. Now, with the trim Galor data that we have, we're going to select pair 1 of each and every output. So you just keep on doing that, control and select. So that is it for the first pair. For the second one, we're going to be selecting pair 2. So all the pair 2s we're going to be selecting for the second file. And yeah, there we have it. Now you don't need to do anything here. You just have to, at least for the time being, we don't, and we have specified all the input. Now with that in mind, what we can do is we can run this tool now. So we'll just click on run again. So when the data is still running, let's do something. We know that with the next step of the process, we're going to be counting and annotating the reads, right? So we need a GTF or a GFF file to do that. So mapping is just it's just a single step process. Now for counting, however, somehow Galaxy does not provide the GTF file for the reference. So we'll have to manually download it on our own. So while the data is running, what we're going to do is we're just going to open Google, and we are going to look for gen code, GNCODE. So just search for gen code, and obviously it'll be the first search option. Now what you will do is you will go to human here. You will open that, and you'll be presented with a list of options here. So remember, we're interested in GTF files or GFF files, right? Now looking at the description, the one we want is this one, the basic gene annotation. So this is the main annotation file for most users, and that is exactly what we will be using. So after opening GenCode, human, basic gene annotation for all chromosomes, the main annotation file. So in GTF, you're just going to right click and you're going to copy the link. Okay. Under basic gene annotation, and on the GTF file, you're going to right click and you're going to copy the link to this GTF file. Now, once that is done, what you will do is you will go to upload data again. So you're going to go upload data. And this time, what we're going to do is we're going to paste or fetch data. So you have three options here. What we're going to select is the last one that says paste or fetch data. Okay. Now you're presented with an option where you will directly paste that output. The link that you copied will be pasted onto that uh, option. And what you can do is you can rename the file. You can just call it something like human encode, anything you wish. You can rename that. And once that is done, you just press on start and it'll upload it. It'll take it directly from that link and it'll be imported into your history. Well, I hope that the file has finished downloading. In the meantime, I guess the alignment files are already generated. So in case it's not, let me just show you what it looks like. So by right-clicking and opening one of the files, you will see what the output looks like. So here is a sample of the output. So this is a BAM file. You can see the header here. These are all the headers. And if you keep on scrolling down, this is the actual part where this is the actual reads. So the main result is this. So the name of the read and the reference, you have your cigar score, you have the alignment, and then you have all these scores, just like the one in FASTA. And then you have a set of other things that if you're interested, you can look it up because SAM-BAM tools, they have a whole lot of information. It's very comprehensive. It has details on what all these output means. And generally, when we're doing this kind of analysis, you're not really bothered with the type of output. I mean, you don't exactly need to go through all these in detail, but if it's something that you would like to do, of course, you can always go ahead and look into it. So in the meantime, I hope that the gene code file has been downloaded. You can take a look at the format of that file. We've already discussed this, but you can still see that this is the format. So the seq name, it contains the chromosome number. So chromosome 1 has a lot of genes. The source is from Havana, and you can have multiple sources, not just one. We can see that this one codes for a gene. It starts from here till here on chromosome 1. Then we have the score, we have the strand. Plus means it's forward. Minus, obviously, it means reverse. That's the alignment of the gene. And then we have the frame, the attributes. The attributes is the part which we are concerned with. So it'll give you the gene ID. So this is the ensemble gene ID. And we have the gene type. It's a long non-coding RNA. We have the gene name and some other stuff as well. So, I mean, the kind of entries here will vary. So if you want to know about each one, again, you can look it up. The internet will obviously give you much more information on that. So now let's move on to the next step, that is counting and annotating. So we're going to be using a tool called feature counts, right? So feature counts, you're going to select that. Now you'll be presented with this screen here, and we're going to again select multiple data sets. And directly we're going to take the yeah, and speaking of which, it's always a good idea to label your files. I'm just going to show you how that's done once we have uploaded this. But yeah, I'm going to take the high set output, all the align BAMs. I'm going to select everything. All right. Now, once that's done, there are some certain things. Of course, I'm sure that everyone's taking notes, but this is something you'll have to pay attention to. Gene annotation file, the one that we have downloaded. So under that option, in the drop-down menu, you will select a GFF or GTF file from your history. Okay. And automatically the tool will select the GTF file that is already present in your history. So in my case, I renamed it as human gen code. So that's what I'm getting here. If it's not there in the list, you can obviously look it up, but generally it should be automatically selected. So once that is selected, what you're going to do is under the GFF feature type filter, I'm just going to keep it as exon because either way, exons are a part of gene, right? So I'm just going to leave that as exon. Over here, however, we're going to change something else. In GFF gene identifier, it says gene ID, right? So if you run this in default, if you just keep this as gene ID, what will happen is it'll select the character that comes right after gene ID, which means it'll select the ensemble ID. Now, some people may be happy with that. Maybe they're okay with having ensemble ID as the identifier. But for most people, it becomes very confusing because generally we identify features with the name of the gene. Like here we have DXX something, right? So gene names are the ones that we are most familiar with. So instead of gene underscore ID, we're going to change it to gene underscore name, right? So if you don't want to go and look up on your GTF file again, even here, uh, the tool itself, it has specified what kind of attributes that you can change the names to. So here they have given gene ID, gene name, gene source, etc. So I'm going to select gene name so that after I run feature counts, instead of IDs, which we can't read, and which we again have to go through the pain of converting it to uh, gene names, we'll directly select the gene name and use that as an identifier. Now, after this, there are some small parameters we have to be careful about. So on feature level, we're going to turn that on. We're going to say yes, use feature level, right? Now, after that, um, in the output format, this one is already set to multi-QC, DE-seq, HR, Limavum compatible. So that is fine because we'll be using DE-seq for our analysis. So we'll leave it as such. Now we're going to be creating a gene length file. This is perhaps the most important if you want to perform, analysis beyond the gene expression um, analysis, which we will be doing. If you want to do gene ontology analysis, for example, you will need a gene length file, especially while working within Galaxy. So we're going to create a gene length file, which will tell us exactly the length of the sequence which is aligned to that feature. So we're going to say yes. And after that, since the input that we're using is a parent, we're just going to say yes. paired but still count them as if individual reads. So the middle option. All right. And with that, what we can do is we can run the tool now. Now there's a thing that I have missed here. You'll see that the output from HiSet. Now the input that we provided initially, there were 12 files. Now it's been converted to 6. Why is that? Well, the reason is that, like I said before, it's not two files each. It's just a single file, but it has been divided into read 1 and read 2. So coming from both the directions. What HiSet2 will do is it'll find the pairs on its own. So normal replicate 1, read 1, and normal replicate 1, read 2. It'll merge them together and it'll create a file. And the output will be one single file. That is why we have HiSet. I've renamed it on normal one. And that's how we have only one single file here. So that's one thing. The second thing is, if you look at the output here, if you click on any one of the HiSet results, you will see something that can be scrolled here, right? So what it'll do is it'll show you what the alignment rate was. So how much percentage of your sequence has been aligned to your reference genome. Now, the first statistics, it says that this many, that means there we have this many reads, and out of that, this many reads, 100% were paired. And we already know that these were paired and reads. The second one says, yeah, this many align concordantly 0 times. That means 7.44% did not align to the reference genome. 87.76 align exactly once. And around this much, that's 4.8% align roughly greater than one time. So the overall alignment rate, if you calculate, it's going to be 87.76 plus 4.8. So overall, it's okay-ish because what you see here is that the total alignment rate is greater than 90. So that's pretty good because if it falls below 90, if it's around the 80-ish mark, then that's going to be a problem later on because that means that the genome that you have aligned is actually not aligning to the sequence. That means you're either using the wrong genome for as your reference. Maybe there's something wrong with your sequence overall. Maybe there's contamination. Maybe it's mixed up with some other genome. So those are things that you should know. You have to check the alignment rate and see that it's at least up to 90 to 95% should be aligned properly. Once, more than once, doesn't matter, but you shouldn't get a message saying that 15% of the gene were aligned 0 times. So that's a big no. So let's take a look at the feature count output. This is what you will find. You'll have the entire list of genes that are present in the human body. You can scroll this down even further. And here on the other side, you will see height set on normal one. So this is just for the first file. For the second file, you can keep on checking for each of the output. But actually, let's not do that because we already know what we are looking for, right? Now, after we do this, yeah, so 0 means this gene has 0 read count. So that means none of the genes, none of our reads are actually aligning to this gene. And you might see that there are a lot of zeros, right? Zeros, ones, mostly zeros. I assure you that there are more than zeros here because if you try and sort the values by their largest digit, you'll find some few thousand here and there. The reason is because, remember I told you that we are using the data set that was taken only from chromosome 22. So imagine these genes are not in chromosome 22. So obviously there's no way you will be getting any reads here in that case. That's the reason why. So you will get similar output for each of the samples. So for normal sample 1, sample 2, 3, 4, etc. And besides that, you probably also have a feature length file that is generated. So this is the feature length file. This is what it looks like. We have gene ID, same thing again. This time we just have the length. So how much of a read was aligned to this? So it'll just give you a rough length of that as well. So if you think about it, this says 3055, but we know that our reads are only 155 in length. How did this 3055 come about? Well, it's because we're not just finding a feature of one single gene that aligned to this region. It could be multiple, right? So that's what it'll give you. And also, it's not just the length of the file that is actually present. It'll give you the length of the entire gene that actually, because remember, DX1, this gene doesn't even show up in our list, right? But it's showing here because this is the overall length of the gene. That's simply what it is. So now that we have our feature count output, finally, we can perform differential expression analysis. So all these 3 steps are done. With all these output, we will be merging them together and we are going to get our output here. To do that, we're going to search for DESeq2, right? And we're going to click on DESeq2. All right. So I hope you've opened up the tool here. The first option here, it says factor. Now the factor, you can just change the label here. It says factor name by default. You can change it to something like, because we're dealing with cancer cells and normal cells. So you can say cancer underscore versus underscore normal, something like that, or treatment versus normal, anything you want. So once you have labeled that, you will go to factor level. Or like, so we're going to specify a factor level. Typical values could be tumor, normal, treated, control. This is the part I was talking about with that question earlier, where people get confused which file to take. How do you know which one should go first? Is it the treatment? Is it the control? So we will be selecting the counts. Remember that counts, not the length. And we're going to select treatment here. Make sure it's not controls and make sure it's not length, it's counts. So feature count on blah, 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 something counts, right? And it has to be the treatment, not the normal. Normal will go to the second factor level. Here you will specify feature count on normal. So what this will do is you're going to be comparing the genes that are present in our treatment file or the cancer sample against the normal. So anything that is expressed will be shown with respect to the cancer cell and not the normal cell. So if you've selected that, what you will do is we're going to change some few things here. We know that the count data is from feature counts. So we have already selected that. If you're using something else, if you're using TPM values, so that's just a you know, Callisto, Sailfish, Salmon. These are all different tools for quantifying your read counts. But since we're using feature counts, that's what we will be doing. You can check out the other parameters if you want. It doesn't really matter because right now we won't be using any of those. And in the output option, sometimes you will see all these output sample size factors, normalized counts. Sometimes people like to use normalized counts, especially if you're doing analysis like GSE, gene set enrichment analysis. Right now, we won't be doing that, so I'm not gonna select any of that. I'm just gonna say generate plots for visualizing the analysis result. But for if you want to do RNA-seq for your own study, then it's always a good option to turn the normalized count output uh, and generate it. So once you have selected this, let me just rephrase that again. You don't need to change the names. If you want, you can do it. I've just changed it here. You're gonna select only the treatment files or the cancer files or the file you want to compare against and place it in factor level number 1. And whatever the control is, be it normal or whatever it may be, that'll go to factor level 2, right? And once that is done, you will run the tool once more. Now you'll be provided with two output. So one will be a text file or a tabular file. The second one will be a plot in the form of PDF. Now this is the actual output that we were looking for. So let's just discuss what the output is. Yeah, so this is something that you will see here. The table, you can just click on the eye icon and you can visualize that if you want. So the first column that we see, it's the gene ID, obviously, and we label that using the GTF file. So at least we're presented with the gene name now and not an ensemble ID, which we cannot understand. The second thing that we see is the base mean. So base mean, it's nothing but the average of the normalized count values. Remember, we normalized all those files. So this is just a normalized average of the normalized count values, right? The important thing here is the log to fold change. Okay, so log fold change, I think I've already explained what it means. So this tells you how much the gene expression seems to have changed in our cancer sample in comparison to the normal one. That means this gene is upregulated or overexpressed in cancer type, and whereas it's not expressed or it's down in normal. So that means this is upregulated, right? That's what it is. And then we have something called the standard error. It's just an error, basically. That's what it is. You don't have to worry much about all this because every time you calculate log fold change, and it's a statistical process, right? So you're bound to have some margin of error. So this is what the standard error is. Wall statistics is basically the statistics which we have applied in order to generate the p-value. And the wall statistics, if you do the calculation, it's nothing but it's what do you call? You just divide the log fold change by the standard error. So you just take this value, divide it by this, you'll get this. As simple as that. Then finally, we have the p-value and the p-adjusted value. That is the false discovery rate that we have discussed earlier on. And yeah, that's basically it. So these are 0. If you scroll down a bit further, yeah, this will seem more reliable because having a p-value of 0 sounds almost, kind of fake. But this seems to be reliable. You can see that this is a really good p-value, and not just the p-value, the p-adjusted value is the one that you should be looking at. So having this low p-value, it's probably very much correct. And you can check the expression also. So gas 2L1, that's the name of a gene. It's really expressed because you can see that 5, generally we say that anything that is greater than 2, it's expressed like by a lot. This one is pretty good. This one is pretty good too. These ones are pretty good as well. 6.4 overexpression and minus 6.7 underexpression. If you check their p-values, sorry, the FDR again, you can see that they have very, like, the numbers are quite small. So obviously we can say with confidence that these genes are expressed and these are not false positives. These are actually expressed. So that's what the table tells you. And this is the output that you will be using for most of your analysis. So what you can do is, of course, you can download the data. You can just click on this save icon, which you shouldn't do now, but you can do it later on if you want. And you can just manipulate the data using, Excel sheet or something. You can sort the values. Anything that is greater than 0.05 can be deleted, and anything that is between -2 and 2 can also be deleted, so that you are left with only the ones that are truly expressed, so as to say. Now, that's pretty much it for the output. Let's take a look at the plots as well. So the plots here are quite interesting. Yeah, here it is. So you don't really have to, understand each and every detail of what the plot means, but I'll just give you a general idea of what it is and what it represents. So I'm pretty sure most of you are familiar with PCA, principal component analysis. So it's a statistical technique, again, which is used to reduce the dimension of the data. So these are the principal components, and it explains the majority of variation in the data. What it does is it projects the samples onto these principal components, and it helps us in identifying the cluster of samples based on the similarity of their expression profile. So we see here that feature counts on treatment 1, 2, 3, that is our cancer data, are clustered very close together, right? That means their expression profile is very similar in nature, whereas the normal ones are scattered here and there. But the point of the fact is that these two are clearly separate. There's no mixing of these. So it looks like we have a good sample in our hands. Next is the sample to sample distance. So it's just a heat map based on Euclidean distance of samples. Uh, Again, this is just a way of visualizing the similarity or dissimilarity between RNA-seq samples. This is again based on the expression of gene profiles, and it's somewhat similar to the PC output that we have. Now, this heat map, it represents pairwise differences between samples based on their expression. So it can help identify clusters of sample that are more similar to the other one in terms of their expression, or more dissimilar. So if you look at PCA, it just tells you that these things are clustered in this region. It doesn't actually tell you if this one is similar to this one is similar to this. What is the connection of this with that, right? So this one actually does that, because if you look at the treatment based on how dark the color is, you can say with confidence that these are similar. So this one, these two are the most similar here. So treatment 1 and 3, 2 is again similar to these. And again, these two, the normal ones, are completely on a different level. And what is the correlation between them? Light blue, which means there's almost no similarity between them. That's what it will tell you. Now, this one is a little tricky because it's hard to explain this without getting into all those estimation of size factors and fitting of shrinking of genes, dispersion estimate. I'll try my best to explain what this is. So on the x-axis, we have the mean of our normalized counts. So remember all the genes, the reads that we normalized before. So this is the mean of that count. And we have the dispersion, which basically means the variability in the data. So what we do here is this is the curve fitting I was talking about, and here we have it. We have the genes, the things that are um, the black dots here. We have the fitting and then the final fit data. Now, whether a gene is significant, because we've seen that log fold change and adjusted p-value, these are the things that we look into, right? But besides that, um, these are not the only criterias which can through which we can say that a gene is significant or not. Okay, so we can quantify this as the dispersion. So each black dot, it represents dispersion of one gene. So every black dot is one gene. And like I said, the red line here is the line that is fit, that was created to fit the data, the black dots, the data. And then dispersion squeezes this. Uh, the final dispersions are the one in blue. So it's just squeezing it towards this line. And usually the dispersion is highest at the low counts and levels off towards high counts. So it always ranges from 0 to 1. So the more dense and more stable it is, it'll be towards the high, and likewise. Again, it's up to debate, but I feel that this is not much relevant because sometimes the plots can change. And this one, to be honest, looks quite weird because this isn't how the dispersion estimate actually looks like. But since we're using only a small portion of a genome, we're using only chromosome 22, which again is quite a small chromosome. So we're getting a pretty weird shaped dispersion here. Nothing wrong with it, but just a general comment. And then we have the p-value histogram. So what this will show you is the p-values on the x-axis. So p-values obviously from 0 to 1, 0 being the best, and of course 1 being the worst. And then we have the frequency. Frequency, basically that's just the density of our genes. So you can see here 0.05, it's pretty close to 0. So these are the regions of treatment versus normal, where most of our differentially expressed genes are. The rest we can neglect it. It doesn't matter if the density is more or less, because anything that is greater than 0.5 is discarded, or we are not really interested in that. So this is just a plot that shows you what it looks like. And again, this also doesn't normally look like what it does, but due to the fact that we have chromosome 22, it's a little weird. And then finally, we have the MA plot. Uh, what it does is, again, it represents each gene with a dot. On the x-axis, again, we have the mean normalized counts, and then we have the log fold change. So anything that is above 0 are overexpressed, anything less than 0 are underexpressed. So again, just a way of visualizing it. But generally, when we talk about differential gene expression analysis, or DEGs as we call it, DGE, yeah, that's right, DGE, uh, we go for something called a volcano plot. Volcano plot, you must have seen it in the workshop earlier, you must have seen it in several papers. So we're going to try and plot one of these. So let's go to tool once more, and let's just search for volcano plot. All right. Now we're just going to click on that tool. This one is quite straightforward. We're just going to say, take the DE-seq result file on data, whatever it is that's in your system. Or if you haven't generated, that's okay. I'll just show it to you. So you select the tabular file that was generated, and you're going to specify where the adjusted p-value is. For a fact, I know that the adjusted p-value is in the last column. That's column number 7. So I'm going to say column number 7. All right. If you want to specify the p-value, the raw p-value, you can say 6, but I'm not going to do that. I'm just going to go ahead and select the log fold change. So log fold change is on column 3. So I'm going to select that 3. significance threshold, anything that is, that's the fold change, 0.05 by default, fine, we'll keep it at that. And log fold change threshold. So log fold change, let's keep it 0 for the time being. And we don't need to change any of this. Of course, you can always label the plots if you want. But once you're done with that, so the important thing is to select the FDR rate, that is column 7, and fold change on column 3, that is it. And then you execute the tool. This actually shouldn't take much time. Regardless, let me just show you output. So this is what the actual output will look like. Okay, so here, what we are seeing is the gray ones. That means these are not significant. They either have a very suboptimal fold change value, or their FDR rate is way above 0.05. So these are not significant. The red ones are the ones that are upregulated. And then, of course, the blue ones are the ones that are downregulated. You can see plenty of genes that are shooting up way past the border here. That means you have some like genes that are really high in expression, the values probably up to 11 or minus 11 fold change. That's what this is all about. So yeah, this is basically all there is to RNA-seq analysis. Now, the steps that follow this, it depends entirely up to the kind of experiments you want to perform. Like I've already explained about gene ontology analysis, right? So these are all the things that can be done. Now, because time is of the essence and we're almost out of time, I have already calculated some outputs which I will be showing you right now. This is a gene ontology of biological process. So with those genes, what I did was I simply used, remember when we calculated gene length from features count. So using that gene length, I specified that. And by using the expression of the gene, the log fold change, and of course the FDR value, I calculated this. And the plot was, of course, generated by, you can write this down. The name of the tool is called GeoSeq. GeoSeq. So it has a wide range of application here. In fact, let me just open that here. Yeah, you can see this. So for this, you'll need a differentially expressed file and you'll need a gene length file. And once you do that, you can select what you want to do. Of course, human genome 38, we're using gene symbol, and then select what you want to see. If you want to see where inside the cell the gene is present, you will select cellular compartment. If you want to see what biological process it's involved in, and so on. And then you also have option for KEGG, which KEGG, as you know, Kyoto Encyclopedia of Genes and Genomics, I guess. So you'll see like several pathways that are that scientists have discovered and mapped till now. So you can find out exactly where your gene is affecting in one of those pathways if it's present. And there are several ways of doing this, of course, but back to the output. So here we see that there's a lot of genes with very low p-adjusted value, which is good, but the count is quite small. They're involved in cytoplasmic translation. So this is for biological process, by the way. I just generated one plot, and likewise, you'll see how many genes are involved where. And if you look at the other output, it'll also give you a list of exactly what those genes are. You will have, sorry, yeah. So cytoplasmic translation, right? And then you'll also have a list of genes that are involved in the process of cytoplasmic translation. Let me see if I can open that up. Okay, I seem to have deleted the output for that, but I'm pretty sure it'll give you a list of the genes as well, not just the figures. Something like this. See, here you can see all the genes that are involved in this specific. This is the KEGG pathway, by the way. So using this again for KEGG, you can generate it using GoSeq, just like I did. Uh, Once you do this, you can create some really nice looking pathways as well. So just by selecting the KEGG pathways that are present, I have used a tool called PathView. You can write that down as well because PathView, I believe, you don't necessarily have to use it within Galaxy. You also have an online GUI version of PathView besides having an R tool as well. So this was originally an R tool, but you can find it online and you can use it as a standalone tool. So you can just list all the pathways that are available. I just made a list already of KEGG pathways, specify the species, provide the gene data, and once you do that, you'll get an output like this. And if you open one of them, look, this is what you'll see. So the information we have here is it'll give you which genes that are currently in your list of expressed genes are present in this pathway, the leishmaniasis, one of the diseases, and whether it's overexpressed or underexpressed. So this is a good way to start your analysis because generally when we perform DGE, gene ontology is the one that usually follows immediately, and we often have to look at pathways as well. So you get this kind of outputs, and everything, each one of these were done from within Galaxy. So uh, yeah, that's about it. Uh, I think that sums up the entire workshop for today.